Proteins are the workers that run our cells. So for example, if we talked about photosynthesis and we talked about chlorophyll. Remember, chlorophyll was the pigment inside the thylakoid membranes that actually absorbed sunlight. All pigments are proteins. So chlorophyll is a pigment, it's a protein. Melanin is a pigment, it's a protein. Okay, guys, I need you to stop talking. These are all the worker molecules in your cells. Okay, so the little penguin is right. DNA gets a lot of glory, but proteins are the ones that actually do the work. Okay, proteins are the ones that run your cell membranes. Remember we talked about cell membranes and all the proteins that were in that, transporting stuff in and out of the cell. Proteins are the ones that hold on to your oxygen and transport it around your body. Proteins are the ones that help you digest your food, so on and so forth. Insulin is a protein. So insulin is going to help you control your glucose levels. So proteins are the ones that run everything. Okay? DNA is in the nucleus. It never leaves. Never, ever leaves. It is highly protected. Okay, so we keep it protected in the nucleus. So there's a reason for that. DNA is, like I said, the most important molecule in your body because it codes for those proteins. Now, when DNA is messed up, then that code is messed up. And when that code is messed up, then the proteins get built wrong. And we don't want that. We don't want proteins to be built wrong because when that happens, that can cause all sorts of dangerous problems in your body. For instance, if I have a code for DNA that codes for hemoglobin, hemoglobin is the protein that sits on my red blood cells and transports oxygen throughout my body. Now, I don't want hemoglobin to be built wrongly. If hemoglobin does not work, how do I get oxygen from my brain to my toes and back from my toes to my brain? Does that make sense? So we don't want DNA to get messed up. That being said, typically, depending on wherever you come from or wherever your ancestors come from, if your ancestors come from a place where there's a lot more UV rays, for instance, closer to the equator, then they would have adapted or evolved to have more melanin surrounding the nucleus. That melanin is darker and it shades the nucleus so as to protect the DNA because UV rays destroy DNA. So the darker you are, the more melanin you have, the more your DNA is protected from UV rays because you or your ancestors come from a place where UV rays are higher. Okay? Yes, sir.
So, could you take DNA out of somebody? Like, could you, the scientists do that?
Absolutely.
Really?
Yeah, you can take DNA out of people. People do it all the time. So, whenever you want to do a paternity test, you just swab the inside of their cheek, so their cheeks get some of those cells, and you take the DNA out and you compare. Of course you can.
I bet
Huh? And are chains of amino acids, and they are made in the protein factory called the ribosome. Now, ribosomes, not necessarily, Esther. Typically speaking, if you get sunburn, that's just the destruction of the cells, the skin cells on the top layer. You have so many layers of skin cells. In addition, one of the issues is that if you are constantly in the sun, you could possibly destroy DNA, especially if you don't have enough melanin to cover up your nucleus. And so individuals with fairer skin are often told not to be in the sun too often. But typically speaking, you won't destroy you'll destroy the entire cell, the layer of cells on the top, but you have so many layers that you should be okay, it'll heal. Good question. So proteins are not in the nucleus, they're on the outside, remember? They're either sticking on the rough ER or they're hanging out in the cytoplasm. Okay, so that's where, I'm sorry, ribosomes. Ribosomes are not in the nucleus. So that becomes the issue. How, if all the information is in the nucleus, all of that information is in the nucleus, and proteins, the ribosomes, are out in the cytoplasm, and that's where we're going to build the proteins. How do we get the information out of the nucleus to the cytoplasm to build the proteins? That's the problem. Because DNA, like I said, never leaves the nucleus. So how do we get DNA's information out of the nucleus and into the cytoplasm where we could build some proteins? Cue in messenger RNA. Write that down. So it is messenger RNA that copies DNA. So it copies, so we get a copy of DNA in messenger RNA, and then the messenger RNA leaves the nucleus, goes out to the cytoplasm, finds a ribosome, and begins to help build the proteins. So messenger RNA is almost like a little thief that breaks into the vault, copies the information, takes it out to the cytoplasm in order to build the proteins. So what? Yes. We're about to move on if you're running out of space. Don't worry. Okay. Central dogma theory. So that little space that you guys have right there, and you're like, what is dogma? I know. So you should have titled it central, like center, central, and then dogma is spelled D-O-G-M-A, dogma, and then theory, central dogma theory. This is what this is called. The central dogma theory states that DNA is copied into mRNA. That process occurs in the nucleus, and it is called transcription. Hey, Jordan, can you do me a solid and close that door? I appreciate you. Thank you. I was like super loud. Oh my gosh. Okay, so I want you to write DNA and then draw the arrow, write mRNA, put transcription above the arrow. Make sure you include that this occurs in the nucleus. So this happens in the nucleus. Now, mRNA has copied DNA. We call it transcription. The last process, part of the process, is to actually make the protein. Go out to the cytoplasm, find a ribosome, and start making protein. Now, that process is called translation. So today we're going to knock out transcription in that process, and we're going to knock out part of translation. Thank you, Jordan. Under the central dogma theory. That's where this goes. You didn't miss too much, baby. This goes under central dogma theory. We'll get you caught up on that. on the background just a bit, okay? DNA, arrow, mRNA, arrow, protein. Make sure you include that DNA to mRNA is called transcription. It occurs in the nucleus. And then mRNA to protein is called translation, and it occurs in the cytoplasm. Okay, so now we need to compare DNA and RNA. These are both nucleic acids. Remember that, right? And also ATP is a nucleic acid, right? So those three are nucleic acids. And being nucleic acid, the monomer for nucleic acids is a nucleotide, right? So we know that a nucleotide is a phosphate group, a sugar, and a nitrogen base. So every nucleic acid has a monomer nucleotide, and the nucleotide is made up of phosphate, sugar, nitrogen base. That having been said, I want to just kind of remind you of that. Let's go ahead and compare DNA to RNA. Okay. Okay, starting with DNA and the D in DNA. The D in DNA stands for deoxyribose sugar, because the sugar in the DNA nucleotide is called deoxyribose. And in fact, all of DNA is deoxyribonucleic acid. That is its name. So deoxyribose sugar. Can you guess what RNA might be? Not deoxy, but just ribose sugar. Does that seem familiar? The ribose sugar?
Yeah.
Where did we see ribose sugar before?
ATP.
We saw ribose sugar before in ATP.
Yeah.
Remember, ATP had a ribose sugar. Now, DNA has nitrogen bases. We know this. The nitrogen bases for DNA are G, C, A, and T. And we know that T and A bind and G and C bind. Now, RNA does have nitrogen bases, yes, but it's slightly different. There is no T in RNA. There's no T in RNA. So instead of T, we have a U. So that A now with U. So if you see an A on the DNA side, you must put a U on the RNA side, because RNA does not have, guess what? T. There is no T. Now that's going to bother a whole lot of you guys, because you're going to be like, nah, Miss Johnson, I don't like that. And that's okay. It is what it is. So previously when we did our little foldable, that foldable was kind of easy because we went A, T, G, C all the way. Now you have to start really considering what it is that you're making. Are you making DNA? Or are you making RNA? Because if you're making RNA, you cannot put a T because there are no T's in RNA. You have to put a U. We know that DNA is double-stranded. You drew that. DNA is double-stranded. DNA is linear, or it looks like lines in eukaryotes, but it's circular in prokaryotes. Remember, prokaryotes are all bacteria. So yeah, we're writing all of this down, guys. I know I'm not highlighting, but I'm pretty sure you guys know we're writing all that down. DNA is double-stranded, but RNA is not. RNA is single-stranded. RNA is single-stranded. And while there is only one type of DNA, there are 3 types of RNA. There are 3 types of RNA. We just mentioned messenger RNA. That's the one that steals information from DNA, takes it out to the cytoplasm. We will talk later about tRNA and rRNA. The T in tRNA stands for transfer, and guess what? It transfers. And the R in rRNA stands for ribosomal. Guess what? It makes up ribosomes. Okay, so for RNA, A will bind with U. And then, of course, in DNA, A will bind with T. Now, question. Some of us remember and some of us don't. If A has a double ring, what do we call those nitrogen bases? Only two of them, A and G, are double rings. They're purines. Excellent. A and G are purines. Everybody else is a pyrimidine. T, C, and U are pyrimidines. You just have to remember ags are pure. See, there's not a whole lot of future Aggies in here. Okay. So soon as you've got that down, we're going to go over the process of transcription, which, you know what, it's pretty much as easy as the process of replication, if I'm honest. Not too complicated. Give you guys a minute to write all that down. Next class, we will do another lab like we did last class, but you won't bring your candy. I'll have it for you. So we will, but we'll do it together. We'll do it together. Katie, do you have your hand up? I can barely see it. What's up? What's up? For when you're typing out on the fill in the blanks, Canvas will mark it wrong if you spell it wrong. Usually I can catch it, but I would definitely, if I were you, I would definitely try to spell it right. Usually I can catch it. Sometimes I don't. That's okay. That allows us to work on our spelling, right? Because that's also important. All right, so moving on. All right, so transcription is just the making of mRNA from DNA. So essentially, we're just copying DNA into mRNA, and it looks like this. So transcription, there's our DNA, and we're going to copy it into, guess what? RNA. The DNA strand that we use is called the template or the pattern, and we're going to match bases. A and U, G and C. The enzyme we're going to use is RNA polymerase. Now, previously in replication, we had two enzymes. We had helicase and DNA polymerase. Helicase unwound and unzipped DNA, and then DNA polymerase added the complement. Well, RNA polymerase in transcription does everything. It unwinds and it unzips. So it does everything. All right. So those of you at home, you've already got this picture. You already drew it. Okay, so you should have already drawn this picture. Let's talk about it. So at the bottom, you've got RNA polymerase. Please highlight that. This whole pink part right here is RNA polymerase. Remember, RNA polymerase does everything. It's going to unwind and unzip DNA. Obviously, this is your DNA. I didn't twist it as much as it is on this picture, so bear that in mind. So our 3s and 5s might be a little bit different, but that's okay. RNA polymerase unwinds and unzips DNA, and then it starts to build the mRNA. Okay, so you guys are missing parts of your DNA in your picture, or go ahead and fill in parts of your DNA in your picture. Go ahead and fill in those parts of your DNA in your picture. Do that now. Fill in the other parts of your DNA. This thing here. Today is not a good day for technology. Thank you. And I still can't see it. Nope. There we go. A little better. All right. So here's what you should have. And I will take a picture of this for those of you at home because you can't see this. I know you can't. All right. So here's what you should have. On this side of the DNA, G goes with what? C, and then A with, okay, G with, and this should be G, and then what should be here? T, and then what should be here? A, and then, good. And then this should be T. Should have an A, and then G, and then a T, and then a G. So approximately that's what it should kind of look like. And then of course I've got my big enzyme. What's the name of the enzyme again? Okay, yeah, it is a protein, but what's the name of the enzyme that we're working with for transcription? Now helicase. What is the name of our protein? RNA. What? What is it? Polymerase. Okay, that is the name of the protein. And so now we're going to build some what? Some RNA, right? So let's build some RNA. Starting here on the mRNA, what is the nitrogen base that's going to bind with G? What nitrogen base binds with G for RNA? What do you guys think? C. It's just a C. All right. But for A, it's going to be a U. What about for T? A. And then what about for A? U. And then G. C. Good. You are smart. And then I can't fit the G over there, so I'm not going to bother. All right, so there's my mRNA. I know this DNA over here is 5 prime, so this RNA has to be 3 prime on this side and 5 prime on that side. Okay, so I just built my RNA strand, and that is transcription. Go ahead and highlight your mRNA. Okay, this particular diagram is super important, and I'm going to tell you that I can assure you that this diagram is probably the one that is on every STAR test. For some odd reason, they love this diagram, and it's definitely on your test next week. So you need to make sure that you can say for sure that mRNA is what's being produced, and this process is called what? Starts with a T. What is this whole process called? Transcription. So make sure that you can identify that. A lot of students get it mixed up with replication. For replication, you're making another DNA molecule. Transcription, you're making mRNA. Great. Now, going back to here, I need you to label some stuff. Okay, so what I want you to label, this strand that we used to make the mRNA is called the template. So this strand right here is called the template. Label it. It is the transcribed DNA. And then this strand that we did not use is called the coding strand. All right, turn to the next page, and I want you to practice. I'm going to go a little bit faster now. So what's going to end up happening, since I didn't finish everything, you are not going to do the homework for tonight. You'll do it the next night. That's fine. Go ahead and write this DNA strand on the next page. Your next page where it says DNA. Do me a solid and write them in threes. That's going to become important a little bit later, so make sure you write them in threes. Yeah, just next to the DNA part. Make sure you clump them into threes. Once you do that, transcribe into mRNA. See if you can do it by yourself. All right. You guys tell me, what is the mRNA complement to TAC? Is it ATG? AUG. Remember, every time you see an A, you got to put a U because we're making RNA. There's no T's in RNA. What about GCA? Good. And then CAT? G, what? UA. And then TTA? Good. All right. So I think we get the idea. Okay, so everywhere you put a T, you better go back and erase it and put a U. All right, last part, and I know we're running out of time, but we need to write this down to make sure you're still paying attention. Last part is junk. DNA has some junk in it. DNA has junk. When I say junk, I mean parts of DNA that codes for nothing. There are parts of DNA that code for nothing. So write that down and then draw this picture and label it. Now, the parts of DNA that code for nothing are called introns. But see, RNA polymerase doesn't know that. RNA polymerase doesn't know that it's copying junk, right? So RNA polymerase copies everything into mRNA. So write all this down and draw that picture. Don't worry, you won't be late. Just write all this down and draw that picture. So after RNA polymerase copies DNA into mRNA, and that mRNA is produced, then all the junk gets cut out. And when we cut out the introns, we're going to splice together or glue together the exons. Does that kind of make sense? We'll catch up with this next time.